Bismillahirrahmanirrahim. In this video, we are going to discuss the regulatory mechanisms that are in force during the inflammation. What happens to the cell? What are the various responses of the cells to the inflammatory process that is responsible for further amplification or decrease in the uh, body's response toward the injury. Acute inflammatory response may start after invasion or injury, and this acute inflammation is balanced by secretion of many cellular and humoral mediator, and intensity of the inflammatory response depends on these uh, mediators, the amount of the mediator, how intense is the mediator release, and this is then determining, this will then determine the resolution and repair mechanism. If it is small or low in amplitude, there will be resolution and start of the repair of the process. Or if it is large in magnitude, there will be death of the cell. So positive and negative regulation and modulation of these responses occur at several levels. There will be release of various cytokines, both in mediators that are pro and anti-inflammatory. There will be expression of surface molecule. The intracellular signaling pathway will be activated or deactivated. There are positive and negative feedback loops for these intracellular pathways. Then there is the involvement of the gene expression of the molecules that are involved in this inflammatory process. The onset of the inflammation or inflammation associated damage causes the damage into the host tissue also. It doesn't only uh, damage the invading agent, it also can excessively damage the normal tissue, as happens in hypersensitivity reaction. These self-damaging effects are regulated, or they are kept in check by various mechanisms that are present in the host. Whenever there is tissue trauma or infection, liver primarily and to some extent the macrophages, they start to form acute phase reactant. These acute phase reactant protect the tissue from damage. They aid in clearance of the waste material, hence they regulate the inflammation. Some of the key components of these acute phase reactants are cellular protection factors such as alpha-1 antitrypsin, alpha-1 chymotrypsin, and alpha-2 antiplasmin, and also the plasminogen activator inhibitor. They prevent cytotoxic and proteolytic damage that is incurred by the proteases. Then fibrinogen, plasminogen, and von Willebrand factor along with factor 8, they form the consumed clotting factor to control the inflammatory process. Then there are some proteins that are produced such as ceruloplasmin and haptoglobin. These proteins are responsible for transport of inflammatory factors that are generated during the process. Serum amyloid A and C-reactive protein are immunoagents that are developed in this process. In this, the C-reactive protein acts as opsonin, and it is responsible for degradation, and also control of the process, but it is an important indicator of inflammation. Stress proteins such as heat shock protein and ubiquitin, they serve as molecular chaperone, and they also help to remove the toxic waste. There is also formation of antioxidant, and these antioxidants such as ceruloplasmin, they neutralize the oxygen free radicals. When this acute phase reactants are associated with fever and leukocytosis, this is termed as acute phase response. The other regulatory mechanisms are glucocorticoid. The endogenous glucocorticoid act as natural anti-inflammatory agent, and they are increased in infection and trauma and regulate the immune response. Then there are free cytokine receptors that bind and neutralize excess cytokine, and these cytokines are, when bound and neutralized, they prevent excessive inflammation. And this concentration of these freely circulating cytokine receptors in the serum, it also tell you about the activity of the disease, the extent of the disease. Is it in flaring stage, or is it in quiescent stage? Then there are some anti-inflammatory chemical mediators that are released in this process, and the balance between these two, pro-inflammatory and anti-inflammatory effect, determines the fate of the process. Prostaglandin E2, that are also known as prostacyclin, they have both the pro and anti-inflammatory roles. The soluble mediator, they are responsible for activation of signal transduction pathway. This signal transduction pathway are activated whenever there are complement system activation, there is abundance of pro-inflammatory cytokine, or immune complexes. So they have got a positive feedback effect, and then they cause the expression of pro-inflammatory mediators, chemokine, and adhesion molecule. So this is the example of stimulus response coupling. So the stimulus is causing this effect, that is the expression of the agents that are now taking on the process. So these expressions are stimulus specific, and these are cell specific, that is responsible for the various outcomes and various morphologies of the acute inflammation and chronic inflammation. So the signal transduction pathway that are activated are G protein, TNF receptor, tumor necrotic factor receptor, or JAK-STAT pathway, that is Janus kinase signal transducer and activator of transcription. When they activated, there is negative feedback control also. So when there is stabilization of inhibitory factor kappa B, interleukin 4, 10 and 12, secreted leukocyte protease inhibitor, release of secreted leukocyte protease inhibitor, tissue inhibitor of matrix metalloproteases, that are TIMP, antioxidant enzyme, lipoxin release, glucocorticoid and phosphatase activation, and transcriptional regulatory factors all have got negative feedback effect on the signal transduction pathway that decrease the effect of these, and that will result in decreased expression of pro-inflammatory mediators, chemokines and cytokines. Now the G protein mechanism or G protein signal transduction pathway. Many chemokines, hormone, neurotransmitters and other mediators are associated with this signal transduction pathway activation. There is this inflammatory stimulus that result with the binding of with the receptors, and this will activate the exchange of GDP for GTP and G protein activation, and this heterotrimeric protein is then divided into subunits, and as a result there is activation of phospholipase C. This phospholipase C activation will result in phosphoinositol triphosphate activation or diacyl glycerol formation, and this PIP or phosphoinositol triphosphate, it will increase intracellular calcium concentration, and this intracytosolic free calcium due to this PIP3 will then is responsible for cytoskeleton assembly, adhesion molecule expression, degranulation and oxidative burst, while the diacylglycerol activates protein kinase C, and there is cascade of phosphorylation that result in arachidonic acid metabolite formation, cytokine release and oxidative burst. TNF or tumor necrotic factor receptor pathway is another pathway that is the central to the development of inflammation and its symptoms. It induces tumor cell apoptosis and regulates immune function. It is associated with two proteins. This tumor necrotic factor complex, it is a multi-protein complex that is bind with inhibitory factor of nuclear factor kappa B and nuclear factor kappa B. So these nuclear factor kappa B, whenever it is attached with inhibitory kappa B, it is an inactive form. When this inhibitory factor is detached from this complex, now this nuclear factor kappa B, it is translocated into the nucleus. It functions as a nuclear transcriptor factor and it activates anti-apoptotic pathways, and inhibitors of apoptosis are generated through the transcription factor beta. If this one is in abundance, it can activate the pro-apoptotic pathways as happens in some condition. But the important one is this nuclear factor kappa B. Its activity and this transcription factor is important in the various various mechanisms that are associated with tumor necrosis factor. So this ubiquitination and deubiquitination are key to many diseases. This nuclear factor kappa B is normally bound with inhibitory factor, and whenever it is detached, it exerts its effect. So when this nuclear factor kappa B has got two subunits, when two of its inactive subunit are processed in the proteasome and form their active form, so these nuclear factors kappa B is activated, or it is also activated by degradation of its inhibitory factor. It is bound with this inhibitory factor, so it is in the inactive form. So whenever it is detached from the nuclear factor kappa B, it is now free to work. So these two mechanisms are there for the activation of this nuclear factor kappa B, that is an important transcription factor, and it is responsible for expression of the genes that promote cell survival. JAK-STAT pathway, or Janus kinase signal transducer and activator of transcription pathway, is a direct route from the that is activated by growth factors or cytokines such as interleukin and interleukin 6 and interferon, and this starts a ligand receptor interaction. This ligand receptor interaction of uh, this JAK-STAT receptor, what happens? The STAT proteins translocates to the nucleus where they start gene promotion. So gene transcription starts. The gene expression by the cell is regulated by pathogen and the damaged cell. There are various phases of this. There is first initiation, then activation of the gene, reprogramming of the gene, and finally there is gene silencing. So recognition is through the pattern recognition receptors, PRR, that are, for example, toll-like receptor. They recognize the microbial products, pathogen associated molecular pattern, or damage associated molecular pattern. And this starts inflammatory response and activate intracellular signaling pathway. Due to activation of these signaling pathway, there is activation of gene through transcription factors, nuclear factor kappa B, AP1, IRF, etc. And the expression of pro-inflammatory cytokines such as interleukin 1, TNF, and interleukin 6, along with chemokine and adhesion molecule occurs. So this gene activation amplifies the immune response, recruitment of more cells to help at the site of injury. Then, depending on the nature of the damage and pathogen, there may be switch from pro-inflammatory to anti-inflammatory gene expression. These are interleukin 10, transforming growth factor beta, and lipoxin that control the excessive inflammation, and they prepare the tissue for the healing and also promote the resolution of the inflammatory process. Then downward regulation of the inflammatory genes and restoration of normal gene expression pattern that prevent chronic inflammation and restores tissue integrity. Sometimes there is failure in resolution that is due to phase 3 and phase 4. It leads to chronic inflammation and it contributes in diseases like rheumatoid arthritis, inflammatory bowel disease, and atherosclerosis. Excessive phase 2 or excessive pro-inflammatory activation result in cytokine storm that is seen in sepsis and COVID-19. So there are now targeted therapies against these interleukin 1, interleukin 6 and tumor necrotic factor to control the cell activity by silencing of the gene. So this regulation of the gene is stepwise. There is initiation, activation, reprogramming, and then silencing. So this graph is showing the pro-inflammatory and anti-inflammatory gene expression that occurs during the inflammation. In acute variety, acute type of the inflammation, acute inflammation, there is first within early hour there is peak in the pro-inflammatory gene expression to control the events, and it is at highest in the first 3 hours, and then the acute phase genes decline. Then there is expression of late pro-inflammatory genes. They also continue the inflammation until the recovery phase. The acute pro-inflammatory genes, they are less, they are decreased by the 6 hours, 6 to 10 hour or 8 hour. They controlled the initially damaging agent, and then the late pro-inflammatory genes, they take the place of these. The anti-inflammatory genes start with the pro-inflammatory genes. They are there, but with a very few quantity. They gain height or maximum quantity after 1 day to 2 or 3 days, and finally within 5 to 10 days there is resolution or recovery from the active inflammation that is due to activity of anti-inflammatory gene. If this anti-inflammatory genes are not there, there will be persistence of these pro-inflammatory gene that will continuously damage the tissue and responsible for chronic inflammation and also for various diseases such as the rheumatoid arthritis and other many chronic diseases. The first step in the initiation of inflammation is the recognition of pathogen associated or damage associated molecular pattern that are recognized by pattern recognition receptors, for example toll-like receptor or node-like receptor. These receptors detect the microbial component such as lipopolysaccharides of the bacteria, flagellin or viral RNA, or they detect the damaged host molecule such as HMB1 ATP and uric acid, and these, after detection, trigger the intracellular signaling pathways of nuclear factor kappa B, IRFs, and MAP kinase pathway. After this, there is activation of inflammatory pathway, and this intracellular signaling with the help of transcription factors, it releases cytokines, chemokines, and adhesion molecule. These chemicals, they amplify the immune response and recruit more leukocyte to the damaged area. With this, there is activation of complement system, and C3a, C5a and membrane attack complex are formed, and also the coagulation cascade is triggered that forms thrombin and fibrin. All these enhance the killing of the microbe, blood clotting, and activation of the immune cells. Finally, the immune cells release reactive oxygen species and nitric oxide that increases the destruction of the microbes, but it is associated with collateral damage. This illustration is showing that how these pathogen associated molecular patterns such as bacteria, lipopolysaccharides, peptidoglycans, glycolipids, flagellin, various viruses, small RNA envelopes, fungus, and zymosans that are pathogen associated or damage associated molecular pattern such as cell injury and necrosis products, ATP, DNA, uric acid, proteins, degraded proteins, tumor cells products such as ATP, DNA, uric acid, and tumor protein, and extracellular matrix damage material that is recognized by pattern recognition receptors that are toll-like receptors or that are node-like receptors. They activate the innate immune response and also the adaptive immune response. And with this activation, they are responsible for continuation of the immune response. So if these pattern recognition receptors overactivated, uh, they can lead to chronic sort of inflammations or some autoimmune diseases such as lupus erythematosus and rheumatoid arthritis. They are also linked with excessive activation of pattern recognition receptor is also linked with sepsis and cytokine storm. Toll-like receptor inhibitor, complement inhibitor, and antioxidant, they help to control excessive inflammation. So pattern recognition receptors are the receptors that recognize the damage associated and pathogen associated molecular pattern, and then this receptor pattern recognition starts the process that is responsible for cell signaling, and this cell signaling will result in expression of cytokines and chemokines, and these chemokines and cytokines will make attempt to the for the degradation of the damaging product or damaged product, and there will be production of free radical also. Moreover, there is further damage to the tissue or damage associated molecular pattern are generated because of collateral damage that occur during the oxidative burst and during the release of these cytokines and chemokines and recruitment of further inflammatory cells. All these are associated with activation of complement system and activation of coagulation cascade after the activation of Hedgeman factor, factor 12. So these collateral damage or these addition or these activation will further result in generation of damage associated molecular pattern that is going to further prolong this process. So as long as these molecular patterns are there, the inflammatory process will continue. So this is the interplay between the pattern recognition receptor, how they maintain the inflammatory response. There are four types of these pattern recognition receptors, that is toll-like receptor, nucleotide oligomerization domain, leucine-rich repeat proteins or node-like receptors, cytoplasmic caspase activation and recruitment domain helicases, and C-type lectin receptors. The toll-like receptors are one of the pattern recognition receptors that are found on macrophages, dendritic cell, endothelial cells, and epithelial cells. They detect pathogen-associated molecular pattern, bacterial cell wall components, viral RNA, and fungal carbohydrates, and trigger intracellular signaling pathway such as nuclear factor kappa B, MAP kinase, and IRFs, and that leads to cytokine production. So hence they are responsible for activation of these cells. If there is polymorphism in toll-like receptors gene, they influence the susceptibility to infection and inflammatory disease. Overstimulation of these will result in cytokine storm, septic shock, and chronic prolonged inflammation. So these TLR antagonists are being developed to treat sepsis, autoimmune diseases, and chronic inflammation to overcome the excessive effect of these toll-like receptors. There are many of these and they are expressed on cell surface, many cell surfaces, many of the macrophages, neutrophils, basophils, epithelial cells, etc. And they are numbered as TLR 1 to 11. Node-like receptors or nucleotide oligomerization domain-like receptors are found inside the cytoplasm of immune cells such as macrophages and dendritic cells, and they detect the intracellular bacteria, viral RNA, and danger signals that are present inside the cell. And they activate a multi-protein complex that is inflammasome, and it processes interleukin 1b and interleukin 18. The inflammasome contains NLRP3, a key sensor protein, ASC adapter protein, and caspase 1. So when these proteins are combined, there is activation of the inflammasome within the cytoplasm, and there is production of pro-inflammatory cytokines. These macrophages can die by the process of pyroptosis, that is the mechanism of programmed cell death, and there is also immune activation. The cytoplasmic caspase activation and recruitment domain helicases. Major receptors of this domain, this group, are reg I-like receptor or RLRs, reg1, MDA5 and LGP2, and they are found in the cytoplasm of the immune cell, macrophages, dendritic cell and fibroblast also. They are important in viral infections. They detect viral RNA from the invading viruses and they activate MAV5, that is mitochondrial antiviral signaling protein, that induces type 1 interferon, and alpha and beta interferons are formed and they increase the antiviral defense mechanism. The key mediator of this cytoplasmic caspase activation and recruitment domain helicases are caspases, nuclear factor kappa B, IRF3, IRF7. All these lead to antiviral cytokine activation, and this activity promote inflammation, apoptosis and antiviral immunity. The major receptors that are C-type lectin receptors are mannose receptor, dendritic cell specific, ICAM3, grabbing non-integrin, detectin 1, detectin 2 and collectins. They are found on macrophage, dendritic cell, epithelial cell and endothelial cell, and they detect sugars on fungi, bacteria and viruses, and their activation leads to inflammatory response by means of cytokine and chemokine production, release of damage associated molecular pattern that amplifies the inflammation, the activation of coagulation and complement cascade, and the cellular response that is associated with this one. It enhances the pathogen clearance, antigen presentation and immune modulation. So we can summarize that the pattern recognition receptors, uh, by the damage associated or the pathogen associated molecular pattern, they act via any of these signaling pathways: nuclear factor kappa B pathway, the mitogen activated protein kinase activator protein 1 MAP kinase pathway, or interferon regulatory factor or IRF pathway. So activation of these pathway, these two, the nuclear factor kappa B and MAP kinase pathway, they produce pro-inflammatory cytokines, while the interferon regulatory factor pathway, it induces the pro-inflammatory cytokine and also type 1 interferon, and these activations or activation of these signal transduction pathways will result in activation of transcription factor, and then these transcription factors are then bind with the specific sequences in the gene promoter, and hence various cytokines or products are released. The toll-like receptors that are present on the cell surface, cell membrane, they recognize the DAMP or PAMP, and then they activate the signal transduction pathways, the nuclear factor kappa B or MAP kinase pathway. The endosomes are also responsible for release of type 1 interferon by IRF pathway. So the activation of REG1 by binding to the cytoplasmic viral RNA activates nuclear factor kappa B and IRF3 to increase interferon transcription. Also, the soluble cytoplasmic retinoic acid inducible gene 1 like receptor or RLR can activate nuclear factor kappa B, so they increase interferon and inflammatory cytokine production. So this was about the pro-inflammatory effects. There are some anti-inflammatory, the uh, negative regulators of the inflammation. So the healing of inflammatory process can occur by the elimination of initial stimulus that caused the injury, by the apoptosis of the inflammatory cells when the pro-inflammatory signals are decreased or anti-inflammatory signals are increased, or the debris is cleared from the area. The response, all these responses, they differ in individuals with respect to age, sex, and genetic architecture of that particular individual. There are many mechanisms that are considered as negative regulators of acute inflammation. One is gene silencing and reprogramming, in which the genes are silenced. The pro-inflammatory genes especially are silenced, and there is expression of increased expression of anti-inflammatory gene. So this allows the inflammation process to progress toward the resolution. So pro-inflammatory genes that are expressed are TNF and interleukin 1 beta. So these are repressed in the as a negative regulator of the acute inflammation, and the anti-inflammatory factors such as interleukin 1 receptor antagonist, tumor necrotic factor alpha receptor, interleukin 6 and interleukin 10 expression is increased. Then cytokines such as interleukin 6, interleukin 10, 11, 12, 13, they decrease the TNF production. They help preserve the and uh, preserve the inhibitory factor of kappa B that blocks further inflammatory gene activation. Then protease inhibitors such as SLPI or secretory leukocyte protease inhibitor or tissue inhibitor of metalloproteases. Two, they reduce the tissue breakdown and inflammation. They also control macrophage and endothelial cell activity. Lipoxin inhibit the leukotriene biosynthesis, and glucocorticoids via the hypothalamic pituitary adrenal axis, they suppress the inflammation. They down-regulate the inflammatory gene at both the transcriptional and post-transcriptional levels. Kinases, they break down the bradykinin, that is a potent inflammatory molecule, so reduces the impact of bradykinin, a vasodilator. Phosphatases, so the inflammation involves phosphorylation, that is the turn on signal in the signal transduction pathway. So when there is phosphatase, it removes phosphate from this pathway, and it is a turn off signals for the signal path transduction pathway. Transforming growth factor beta is released during clearance of apoptotic cell, especially neutrophil. It suppresses pro-inflammatory cytokines and promote lipoxin and resolvin production that are pro-resolving lipid mediators. It also uh, promotes phagocytosis of dead cell by macrophages, and it initiates fibrosis as well as healing of the tissue. Thank you for watching this video about the regulation of inflammatory process. If you have any query, please write in the comment box. Thanks.